You know, a lot of times when you, as a scientist, think back to experiments that you've done, even really successful experiments, you realize that at the time that you were doing them, you didn't really fully understand the science involved or the science behind that, what science was going to affect whether you were going to get results or not. And you probably also didn't really appreciate how lucky you were. I'd said I would like to talk a little bit about one experiment that, a wonderful experiment that I did many years ago with Christiane Nusslein-Volhard. It was a time when we were both starting out our scientific careers. We, like many people at the time, this is the late 1970s, were interested in the problem of embryonic development, but at a very basic level. We knew that genes controlled things that you could see happening in the embryos, and we wanted to identify who those genes were, what they did. And there were a lot of different molecular strategies, conceptual strategies that you could use to do this. We decided that we would try a genetic approach. And what that meant for us was that to build on, in a certain sense, to build on our lack of knowledge, and to just randomly mutagenize all genes, knock them all out, and see what happens. And get some estimate, at least, or some global picture of what it was the genes were doing during development, how gene activities were organized in patterns and in sequences to produce this extraordinary phenomena that you see in development. We knew that there were lots of genes, and we knew that understanding the process would probably require that we identify most of them. So we set up in our minds at least this goal that we wanted to identify everything. We wanted to identify all the genes, and this meant a really big experiment. And for those of you who have any sense or experience with working with Drosophila, what a big experiment and looking for lots of genes means is lots of tubes, growing lots of flies, and setting up crosses. Basically, our plan was to feed flies mutagens, establish inbred lines from single males in the first generation, then carry it through an appropriate number of generations to produce inbred homozygous lines that we could collect inbred heterozygous lines that we could collect homozygous embryos from. And we thought about it, and our estimates of what we need to do was something like in the range of maybe 40,000 lines established through many generations. And for us to do that, we realized, and this is actually the first real secret and the unappreciated aspect of this experiment, is that we were starting our jobs in Heidelberg, and we knew that we had to set up lots of tubes where we were going to identify all these genes, and if that was going to be effective, we would have to have ways of almost a mechanized inbreeding of flies. We couldn't sit and set up crosses manually by hand. We would have to have selective procedures that allowed us to manually collect individual, it would avoid collecting individual flies and set up crosses. So actually, the first 2 years of our stay in Heidelberg was involved in this, setting up and designing genetic crosses that allowed us simply to put a fly in a tube, wait 2 weeks, there'd be lots of flies in there, put 2 flies in the tube actually, wait a couple of weeks, and next generation, and by raising it at a different temperature or with some other selective technique, we could just shake the flies out, the surviving flies over, and go through and gradually inbreed the lines such that at the end of the cross down here at the bottom, after 3 generations, we had balanced inbred lines that we could collect homozygous embryos for. Now, it took us 2 years to set that up and to get crossing schemes that would actually work. We set out to do the experiment, and the experiment actually itself only took us 2 rounds of experiments, each of which were less than 2 months in advance. So I guess the first lesson, the idea that if you have a vision and the vision's clear enough, you may still have to spend a lot of work just getting, a lot of the work just getting the assays, getting the procedures right. But then the next thing, and this is the thing that I really wanted to talk about, next number, we did actually start out with trying to make 40,000 lines. As you'll see, we only really carried about 27,000 of them through to the third generation, but by the time we'd gotten to that generation, began to get our first results, we got an important, our first important result. Now, we'd used enough mutagenesis that in the lines that we were looking at, we had about, in these 27,000 lines, we had about 18,000 different mutations. And so that meant that our mutagenesis procedures worked, everything was working fine. Our interest, though, these mutations killed the fly at any time during its life cycle. And what we were really interested in were genes that controlled the way embryos developed. And this is the point, actually, where I think we were extraordinarily lucky. Because what we had to do was now, from all these 18,000, these different lines, we had to collect mutant embryos. We had to collect embryos, look at the embryos that die, and decide whether the gene that was mutated in those lines actually killed the embryos and killed them in an interesting way. And the great result, the thing that we couldn't have anticipated, was that of the 18,000 different lethal mutations, only a tiny, tiny fraction actually changed homozygous embryos in a way that was really meaningful. And we, most of the time, the homozygous mutant embryos hatched and crawled away. It didn't have to be that way. And what would have been really, really horrible for us in thinking back to it is that if every one of those 18,000 lines we picked out, looked at embryos, and there was some phenotype, something had gone wrong, we would never have actually been able to, I think, since there was really only the two of us doing these experiments, sort things out and come up with a coherent picture. So we were really lucky that what had actually happened was that in a way that we didn't fully anticipate, the screen was very selective for mutations that cause phenotypes in homozygous embryos. That is to say, we were selecting for genes that had to themselves be transcriptionally active in embryos, and that in flies, that number is very small. And so the number of mutations was about 586, and actually the number of genes, because if you do complementation tests among those mutations, there are only about, we have only found 139 different genes. So that's a really small number, and that reduced the problem of thinking about development down to 139 components, 139 genes, each of which were producing different phenotypes and could be arranged in pathways and sequences that made, for me at least, the whole process of development almost understandable or structural for the first time. That understanding came from the fact that the numbers are small. The final numbers are small. And the numbers are small not because we planned them that way. The numbers are small because of the peculiar biological features of the way, the way flies develop. It turns out flies develop, embryonic development is very rapid. The mother puts everything the embryo needs that she can possibly put into the egg is put in by the mother. And the transcriptional requirements, certainly early during all these patterning stages, are very minimal. So the fly has stripped down its transcriptional requirements to the rare genes that can't be supplied by the mother, that must be supplied by transcription in this cell and not in this cell. There's genes, the embryo is using transcription to control patterns, to control transitions in time. So we did an experiment, a genetic experiment that was oriented towards identifying phenotypes in homozygous embryos. And because of the biology of flies, the way they worked, we ended up identifying the central set of core regulatory genes whose activities temporally and spatially control the development of pattern. And I think, if I think back to what both Yanni and I, at the time, we thought a lot about this experiment. We, as I said, we planned it for more than 2 years. We actually spent 2 years trying to get the thing set up, and yet we didn't really fully understand how the system worked, how fly embryos developed, until we actually had the numbers and had the results. And I think that's actually a great lesson in biology. And on the one hand, you don't understand the experiment, really, that you're doing until you get the result. And it's really only when you get the result that you actually understand that you actually potentially understand what biology and what the whole system that you're working with is actually giving you. That's true, and so I think it is pretty much, I guess, the nature of science. It's really great to do experiments where you don't know the outcome.